Hi everyone, welcome to our DNA screencast. We're going to cover DNA, RNA, and protein synthesis, and I know the first part of DNA will probably be a review, so I'll go through that sort of quickly. And then I think we left off talking about RNA, and then we're going to pick up with protein synthesis, which consists of two processes known as transcription and translation. So we'll talk about those a little bit later. First, DNA, quick review, like I said, deoxyribonucleic acid. It's composed of nucleotides, and the nucleotides are the phosphate and sugar backbone. Remember, if you're thinking about DNA as a ladder, the backbone would be like the part of the ladder that sticks into the ground, and the rungs of the ladder would be the nitrogenous bases, the A, T, G, or C, and those would be the ones that are running horizontal, the sugar phosphate backbone, and the nitrogenous base. Every nucleotide is going to have that same basic structure. And the four bases that we talked about, the nitrogen bases, adenine is going to match with T, so AT, apple in the tree, and then G and C. So we have adenine, thymine, cytosine, and guanine, and they're only ever going to pair with their match. So C is always going to pair with G, G is always going to pair with C, and a purine is always going to pair with a pyrimidine, if that helps you remember. The purines are adenine and guanine, and the pyrimidines are cytosine and thymine. And a way you can remember that is the pyrimidine, PY, that's a Y right there. Cytosine and thymine both have a Y as well. So cytosine and thymine are pyrimidines, and adenine and guanine are purines, if that helps you remember. A, T, G, and C. That's how they're always going to be pairing. It's called a double helix because that's the name of the shape. It is the sugar phosphate backbone, and then you have the nitrogenous bases acting as the rungs of the ladder. It is also called a twisted ladder, but it's called a double helix, and we know that they pair with the A's, T's, G's, and C's. Here's kind of like a big picture view. If you're looking at the whole DNA molecule, the nucleotides are going to be just this part here that we just talked about, the sugar phosphate backbone, and that's like this blue ribbon looking part here. And then you have the nitrogenous base. In this case, that would be guanine, which remember is a purine base. It's always going to pair with cytosine, which is the pyrimidine base, and you can see that here. So the C's and the G's, they have 3 hydrogen bonds holding them together. And then make sure that you're able to do that. This is something we would be practicing a lot if we were in class together, but just be able to label the parts of the DNA molecule. Like maybe I would, on a test or on your assessment on my WJ, have this S blocked out, and you would have to label that as sugar, the deoxyribose sugar. Or if I'd have this blocked out, you'd have to tell me that that's phosphate. You have the phosphate sugar backbone here, and then maybe I would block this part out here, and you would have to tell me that that's thymine, and you would know that because you know that adenine always pairs with thymine. So if there's a blank here, you would know that would have to be a T. Same thing up here. If I'd block this one out, you would know that cytosine always pairs with guanine, so you know that there would have to be a C there. Now we started talking about DNA replication too. And our cells are constantly growing and dividing. And in order to do that, they have to be able to make copies of the DNA that was in the cell originally. And in order to pass on the DNA or the genetic code to the daughter cells, they have to go through a process known as DNA replication. We also looked at the cells and different cells and how long they last in our body. And different cells are going to last a different length of time. Our skin cells aren't going to last nearly as long as something like our nerve cells. They last our whole lifetime. They're the only ones that don't really divide. That you can look at here and compare your small intestine epithelium, so the outer part of your small intestine. Those cells are only going to last 2 to 4 days, whereas if you look at down here, like I said, the nervous system cells are going to last your lifetime. Fat cells, 8 years. red blood cells 4 months. So you just see a difference in the amount of time that these cells take to divide. And in order to divide, remember, we have to go through that process of cellular replication. Now, the first step in replication, DNA helicase. That's going to be this protein right here. It's going to separate the DNA strand by breaking the hydrogen bonds. Remember, the hydrogen bonds exist between the A's, T's, G's, and C's, in between the nitrogenous bases. The helicase is going to be responsible for sort of unzipping that DNA molecule, that double helix, and that's going to allow the next step of replication to take place. That's going to allow access for the individual nucleotides that are needed for the replication process to continue. This is a video on replication and it's a really good animation, so I'll send that out to you guys. And then after DNA helicase unzips the DNA molecule, another protein called DNA polymerase, that's this guy right here. Those are enzymes or proteins that are going to attach to the newly separated strands of DNA. The helicase is going to unwind, and then the polymerase is going to attach the new bases onto the separated DNA molecule. The enzyme uses free nucleotides and synthesizes complementary strands by reading the original strands. Remember, we talked about the fact that DNA replication, this is a semi-conservative process, meaning that you have the template strand already from the original DNA molecule, and the DNA polymerase is going to use that original strand in order to create the new strand. So you'll still have your same composition of A's, T's, G's, and C's, but remember, we need to replicate it in order to be able to pass on the genes and the DNA onto the daughter cells. When the process is finished, 2 identical DNA strands will have been produced. To summarize, DNA replication, the process by which DNA makes a copy of itself. You may also see it called cell division. They're both the same thing, and it's semi-conservative. Half of the old strand is saved and used as the template. Now let's say we have these strands of DNA here. You have the A, C, G, G, T, T. You can look at that whole molecule and you'll see that there's the complementary base pairs here. A always pairs with T. So if you have an A on one side of the DNA molecule, on the other strand, you're going to have the complementary base pair. A and T, C and G, G and C. Now you can look at this and check and make sure that it's all correct, but it is. Now with DNA replication, remember, we're going to separate those 2 strands and then we're going to add the complementary strand. So now we're going to take this part here. We have the A, C, G, G. Now here we're going to have T, G, C, C again. Now if you look, we created an identical copy. Same thing down here. If we use this as the template strand, the new strand is going to be complementary to that template strand, and what we get as a result is a new copy of the DNA, the original DNA, and we copied it in order to pass it on to the daughter cells. And it's important that each new strand is identical to the original. If it's not, that gives rise to mutations, and we'll talk about mutations a little bit later. And here's another image of DNA replication. I just want to be sure to show you a lot of models of this. Here is the original DNA molecule, double-stranded, double helix. DNA helicase is going to come, unzip this DNA molecule, and the strands will separate. And then you can see the new strands are being synthesized. So DNA polymerase is going to come and it's going to add the complementary base pairs to the separated strands. And then we're going to create the copies. Those copies are going to go to the daughter cells and we're going to get 2 new daughter DNA molecules. And this will conclude the first video. I just want to talk about DNA replication and review DNA in this first video. Next, we're going to start talking about RNA, and then we'll get into transcription and translation. As always, let me know if you have any questions, and I'll talk to you guys soon.